unit 4.4, that is chromosomal aberrations. So the meaning of chromosomal aberration, this is nothing but the changes in the structure and the number of the chromosomes are called chromosomal aberrations. So the changes in the structure of the chromosomes, and you know that the chromosomes contains the genes, and the changes in the structure of the chromosomes which brings about the changes in the number and the arrangement of the genes. And there are four types, you can see. One is called as the deletion, the duplication, the inversion and translocation. First, let us know what is meant by deletion. So it is the chromosomal aberrations where a segment of chromosome is lost. You can see certain segment of the chromosome is lost, and here some genes, because of this loss of segment of the chromosome, some genes which are present on this chromosomes is also lost. So the deletion, there are two types of deletions you can see, namely called as terminal deletion, another one is called as intercalary deletions. First one is called terminal deletion, another one is called as intercalary deletions. So in terminal deletion, what happens is the terminal segment is lost. So you can see the terminal segment which is lost in the terminal deletion. And in intercalary deletion, so as the name itself indicates, the intermediate segment is lost, and this is what is the difference between the terminal deletion and intercalary deletions. So, and you can also see when the deletion occur in one member of the homologous chromosomes, it results in a loop, what is called as a deletion loop. And this is produced in normal homologous chromosomes, and the deletion loop is formed by the segment which is opposite to the deleted segments, and it occurs during pairing in the meiosis. So in humans, the deletion of the segment of the chromosome number 5, it causes one type of diseases, and that is what is called as cry-do-cat syndrome. Next is duplication. Duplication means it is the chromosomal aberration where the segment is repeated. You can see this segment is repeated, and that is it is duplicated, and this is what is called as the duplications. So hence a set of genes it is present that will be doubled. You can see this segment of gene is duplicated and it is doubled. So during meiosis, the duplicated segment, it also forms a loop. So this is what is called as a duplication loop. Example for this is in Drosophila, the bar I is due to the duplication, and this duplication produces a position effect. So here the phenotypic effect produced by the change in the position of the gene or a group of gene, which is called as a position effect. Next type of chromosomal aberration is inversion. So you can see the inversion is a chromosomal aberrations where a segment of the chromosomes break and reunite in a reverse order. It breaks and turns upside down, then reattaches, and this is what is called as inversion. So in inversion there will be no loss or the gain of the genes, but what happens to the genes is it is just rearranged in the reverse order. So it is called as inversion. So the inversion is also of two types. One is called as pericentric inversion, another one is called as paracentric inversion. I briefly explain what is meant by pericentric inversion and paracentric inversion. Pericentric means here the centromere is included in the inverted segment. So this is what is called as pericentric inversion. The centromere is included In the inverted segments, this is called as pericentric inversion, whereas in the vice versa, that is the centromere is not included in the inverted portion, and that is what is called as a paracentric inversion. So the chromosomes with the inverted segment, this also produces the loop, what is called as the inversion loop. Then next is the translocation. The translocation is the chromosomal aberration where the homologous chromosome is exchanged the segments. The translocation produces cross-shaped structure during pairing. The translocation causes position effect and translocation alters the linkage groups. So the change in the number of the chromosomes is which is called as polyploidy. Thank you.